as Lilliglow, and also Amelia Usher Segredo of the Liverpool Lung Project. And thank you again.
It was such an informative lecture. Now it's time to meet Dr. Sara. She is PhD in microbiology and molecular genetics with research interest in cyanobacterial biotechnology. She has strong experience in microbiology techniques and related to selection and screening of broad range of microbes. Dr. Sara, thank you for being with us. Kindly start with your lecture.
Bismillahirrahmanirrahim. Hello everyone. So I'm Dr. Sara and going to present the topic cyanobacteria and microalgae as a source of biofuels. So come towards the presentation layout, and first of all I will discuss the introduction. What are biofuels? Biofuels are basically the renewable energy sources which we obtain from the biomass. And biomass is the any living thing which we obtain from any living material. There are the different types of the biofuels including hydrogen, methane, ethanol and biodiesel. There is a difference between the biofuel and the fossil fuels. Fossil fuels are obtained from the oil, gas, and coal, and which reserves are almost diminishing in the world. So that is why we have shifted towards the biofuels. Now come towards the generations. There are the four generations of the biofuels, and we have a greater focus towards the third generation biofuels, including the biofuels obtained from algae and cyanobacteria, because they have 15 to 300 times more biodiesel than the traditional crops. And with reference to the fourth generation biofuels, they are obtained from the genetically optimized cyanobacteria. Now, a short discussion about the first generation biofuels which we obtained from all the crops which are edible. However, the scientists shift towards the second generation biofuels towards the non-edible foods. And then there it comes towards the third generation biofuels including cyanobacteria and microalgae. The fourth generation including the genetic modification of these cyanobacteria and microalgae. Now, what is cyanobacteria and microalgae? Basically, these are the photosynthetic microorganisms, and the main difference between them is cyanobacteria are the prokaryotes. They are single-celled microorganisms. However, microalgae are the eukaryote. Now come towards the aims and objectives. First of all, these microorganisms were screened from the unexplored areas and the northern areas of the Pakistan, and then we have extracted the lipid as well as the biodiesel, and then we move towards the chemical composition of these biodiesel. We have also detected the ethanol and the hydrogen from these microorganisms. After this, we just move towards the sampling site, and we have done the sampling from the different areas of the Pakistan, and we have shifted these microorganisms towards the lab. So here is the pictorial representation of the isolation of these microorganisms in the lab, because these microorganisms in nature, they are present in the form of consortia, and we when shifted into the lab, so we have to isolate them. There are the different you can say the different types of microorganisms we obtained. Total there were 65 strains. However, 33 were unicellular, 13 were cyanobacteria, and 19 were filamentous. Now come towards the biodiesel production. So this is the step that microalgae, by utilizing the sunlight and water, they grow the lipid bodies within them, and we have to extract these microorganisms from the biodiesel, and then we shift these lipids into the biodiesel. This is the actual chemical mechanism which is happening while conversion of biodiesel and when we obtain these from the microorganisms. So first of all, basically the triglycerides are present within the microorganisms, and when we treat with sodium hydroxide catalyst and as well as methanol, then they come towards into the fatty acid esters, and these are basically the biodiesel. The glycerine, we can also obtain that as a byproduct. Now, come towards the biodiesel productivity. So, different types of fatty acids were obtained from these microorganisms, including the saturated fatty acids, monosaturated fatty acids, and as well as the polyunsaturated fatty acids. Here is the GC pictorial representation of these microorganisms, including the filamentous cyanobacteria, unicellular cyanobacteria, and as well as microalgae. Now come towards the and other mechanisms known as the florimetry. What happens in this mechanism that we utilize the two type of dyes known as the body P and as well as Nile red. So they can bind with the antibodies and the lipids and then they glow and we can quantify them. Now come towards the another aspect of this research including the localization of the lipids. It was performed by the Sudan black staining and as well as confocal laser scanning microscopy. Now, what is Sudan black staining? So, as the name describes, that something black will appear within the cells. So, when this stain will interact with the lipid droplets, then it will appear as blackish in color and we can observe under the microscope. So, here are the pictures showing the staining of Sudan black. Now come towards the another microscopic technique known as the confocal laser scanning microscope. So here the yellow and greenish color are showing the lipid particles within the microalgae and the cyanobacterial cells. The another technique which was performed is the Fourier transform infrared spectroscopy, and this was done to find out the ester functional group within the microorganism which is the main body of the biodiesel. Here are the graphs showing the unicellular microalgae, cyanobacteria, and as well as the filamentous microalgae. And you can see the different peaks of these FDIR at the different wave number. Now, come towards the another major aspect of this research of including the obtain of biohydrogen from these microorganisms. So, hydrogenase enzymes are present within these microorganisms and they can obtain via the mechanisms of different steps like including BG11, BG11 not, glucose in absence and as well as in the presence and absence of the light. So we obtain the biodiesel in all these methods. And here are the pictorial representation of depicting how this process was performed in the lab. Here are the GC peak results showing the presence of hydrogen within these microorganisms. The third aspect of this research work is the production of bioethanol from cyanobacteria and microalgae, and the following conditions were utilized to obtain these bioethanol. Here is the pictorial representation of the peak showing the ethanol production within the microorganisms. Now, come towards the conclusion. Cyanobacteria and microalgae are the source of multiple biofuel productions, including biodiesel, biohydrogen, and ethanol. So, here are the representation of the microorganisms showing which microorganisms showed which type of biodiesel within the microorganisms. So, basically, the source of bioethanol is greatest while the obtaining from the cyanobacteria. Now come towards the advancements that by utilizing the different mechanisms and pathways, we can change the method of production of biodiesel in a greater extent and we can increase the production of these biodiesel. So the take home message is go green with the biofuels. And I am in the end, I am saying many thanks to the IM group of researchers as well as Punjab University Higher Education Commission, the Uppsala University, Sweden, as well as Department of Microbiology and Molecular Genetics, Multan. So thank you very much.
Thank you, Dr. Sara, for your amazing lecture and sharing your research experience with us. Now, I want to invite Dr. Adnan, who is director of IM Group of Researchers and always being a great sport for us, for his conclusive remarks.
Assalamu alaikum, dear valued guest participants and esteemed team. Is the fourth International Conference on Life and Chemical Sciences with the theme From Lab to Life, Emerging Trends in Biological and Chemical Sciences come to an end. I want to express my sincere gratitude and pride for the success of this event. On behalf of the organizing committee, I want to thank our guest speakers, participants, and especially the dedicated team that worked hard to make this conference happen. Our goal focused on providing valuable education and raising awareness through research has come to life through the lively discussion and knowledge sharing during these enlightening days. Organized by the IM group of researchers, this conference provided a common ground for the students, researchers, scientists, and professors in life sciences and chemical sciences to share insights, experiences, and the latest advancement in their fields. The objective we set for this conference were not just met but surpassed thanks to active involvement of everyone present. We successfully encouraged collaboration between experts in biological sciences and chemistry, opening doors to innovative solutions for global challenges, discussions on healthcare, drug discovery, sustainable practices, and the exploration of chemistry, of nature, and biomolecular sciences have undoubtedly advanced scientific understanding, paving the way for future breakthroughs. I want to express my appreciation for the commitment shown by participants in promoting ethical chemical research, environmental awareness, and safety measures. This conference has not only been a platform for an intellectual exchange, but also a catalyst for building a community of responsible researchers dedicated to making positive impact on society. Uh, Special thanks to our distinguished guest speakers whose expertise enriched the conference and to every participant who actively contributed to the discussion. My gratitude also goes to the organizing team whose careful planning and ensured the smooth flow of events. I am confident that the knowledge shared and connection made during this conference will go beyond these walls and make a lasting impact on the laboratories, classrooms, and research institutions represented here. As we part ways, let's carry forward the spirit of collaboration, innovation, and ethical responsibility that defined the fourth International Conference on Life in Chemical Sciences. Thank you all for your unwavering support, and I look forward to future endeavors that continue to push the boundaries of scientific exploration and social impact. Thank you all.